entitled Around the World in Neurodegeneration Drug Discovery. So my name is Alan Palmer and I'll be chairing this session. I'll be joined by leading figures in the field of discovering and developing new medicines for neurodegenerative disorders. And in keeping with the theme, they're all based in 4 different European countries. So I'll come on to let them introduce themselves and their companies in a moment. But first of all, what I'd like to do is to set the scene. So what I want you to do now is to take you back to a sunny day in April in 1906 to a large pathology lab in Munich. And there we can see there's a stocky figure staring down a microscope at a section of neocortex from the brain of a 55-year-old lady who's recently died in Frankfurt. The figure is Alzheimer, and the lady is Augusta Dieter. She's admitted to a psychiatric hospital in Frankfurt a few years earlier when she was 51 years of age. She was disorientated completely in space and time and had profound cognitive impairment. And Alzheimer worked and he cared for her for a few years until he moved to Munich to work in another lab there. And through the microscope, he noticed some 2 unusual structures. First of all, he saw senile plaques, and they'd been seen before, usually in older people. And the other thing that structure that he saw was fibrous structures within cell bodies of pyramidal neurons. And this was the first time these had been seen, and these were then became known as neurofibrillary tangles. And he was very excited by this observation, and he discussed it with his boss, Emil Kraepelin, who's a dominant figure in the world of psychiatry at the time. And he had a major input into psychiatry in terms of classifying different psychiatric disorders. But also he had a major impact in the founding of the field of psychopharmacology as well. But we haven't got time to do that today. So Alzheimer described this case in detail, and he described it as a peculiar disorder of the cerebral cortex. And 3 years later, with more cases described, Kraepelin, in honor of his friend and colleague, named the disease Alzheimer's disease, and that's what we know today. And in terms of the history of Alzheimer's disease and its translation into new medicines, not much happened for many decades until the 1970s. And so what I wanted to do now is to go to central London, the back of Holborn Library, beavering away in labs. There was my PhD supervisor, David Bowen. And he was the first one to identify a specific biochemical change in Alzheimer's disease. He did careful studies looking at post-mortem human brain, matching control samples and Alzheimer's samples for things such as post-mortem delay and age. And he looked at a number of 36 biochemical measurements. Of one of the changes he noticed was a deficiency of the enzyme cholinesterase. And that then caused great excitement. It was confirmed later by Elaine Perry in Newcastle, and also Peter Davis in Edinburgh. And so that sort of introduced a new hypothesis and opened up the possibility of rational therapy for Alzheimer's disease based on correcting the cholinergic hypothesis. And so it was some 21 years later then that the inhibitors of acetylcholinesterase, which serve to increase the amount of acetylcholine in the synapse, were introduced. And there are 3 on the market now. And they have a modest effect on some of the people treated. But what we really wanted is a drug to slow down the disease. So I joined David Bowen's lab just after he made these discoveries, and my PhD there was working on monoamine neurons in Alzheimer's disease, looking at both post-mortem tissue, but also we had access to tissue removed ante-mortem at cerebral biopsy with colleagues in Manchester. So I then went on to work at the University of Pittsburgh. I became a faculty member there. I worked as a principal investigator in the Alzheimer's Disease Research Center, and also co-founded the Head Injury Research Center. I worked there until 1994 when I came back to the UK to join Wyeth Pharmaceuticals, based in Maidenhead. But at the time I joined, Wyeth had bought American Cyanamid for $10 million, or $1 billion in fact. And so as a result of that, things began to change, and they offered me a job in the US. But what I did is I got a group of 4 colleagues from Wyeth, and together with Chris Evans, we formed the UK's first neuroscience startup company called Cerebrus, which in 1999 merged with Vanguard Medica to form Vinalis, which is still on the London Stock Exchange. So I left in 2002 to form another company called Pharmadex, which is based in Hatfield and in London. a contract research organization with my good friend and colleague, Mo Alavijay. I left Pharmadex to form a number of other companies, and recently, last year, formed a new company called Elixa MediScience, which focuses on neurological disorders and their treatment. And neurological disorder, neurodegenerative disorders, fall into a number of camps. You know, we've got the dementia such as Alzheimer's disease, Lewy body dementia, frontotemporal dementia, vascular dementia, movement disorders such as Parkinson's disease, Huntington's disease, and motor neuron disease, demyelinating disorders such as multiple sclerosis, particularly the secondary and progressive forms of this disease. But also what often gets forgotten is eye disorders such as glaucoma, which is a substantial loss of cells from the retina. So at the moment, I've got a number of positions. I'm on the board of One Nucleus, and I'm a proud board member and have been for the last 10 years. I'm on the board of the Brain Tumor Charity, which is a pioneering charity focusing on brain tumors, and also the professional organization, the British Neuroscience Association. I also have a visiting professorship at the University of Reading. So what I want to do now in terms of the next step in the story of Alzheimer's disease and translating our knowledge of the disease into new therapies is to go back to my time in Pittsburgh in 1990, because I remembered it very well. I was living in the north of Pittsburgh, I was driving into work and listening to the National Public Radio, which is their equivalent of Radio 4. I heard this story of a group in London, who's led by John Hardy, that it has discovered a genetic link to Alzheimer's disease. And I know John very well. I knew that he'd been working on the genetics of Alzheimer's disease, and he'd identified a family in Nottingham that seemed to have an inherited form of the disease. And what he was able to show that this was linked to a mutation in the gene for the protein, amyloid precursor protein, a very large protein. And this mutation seemed to be linked to the disease and seemed to cause the disease. So this caused great excitement because the amyloid precursor protein leads to a beta 42, a small peptide, which exists within the center of the senile or neuritic plaques. And what the other feature which was important at the time was Down syndrome. Down syndrome is trisomy 21. APP was existing on chromosome 21. And this then raised and the other factor is that people with Down syndrome develop Alzheimer's disease. They have plaques and tangles in their brain. So this was forming a very neat story as to what was the cause of Alzheimer's disease. It looked as if it was attributable to a mismetabolism of amyloid. And John Hardy put out the hypothesis, the amyloid cascade hypothesis, around that time. And that then brought a number of pharmaceutical companies into the field to try to target this, to find a treatment to slow the course of the disease down. That was 30 years ago now. I said it was 21 years for the cholinergic drugs to come from the initial discovery. So that hasn't happened. And it hasn't been for a lack of effort, because tens of billions of pounds have been spent on trying to find drugs to slow the process of the disease down, targeting amyloid. And over 400 clinical trials have failed to show any clear efficacy signal. So the investors so there hasn't been a very good return on investment, and investors have, to some extent, been put off of brain disorders, particularly Alzheimer's disease. But even so, the clinical need remains high, and it's getting higher because at the turn of 2010, the baby boomers, the people born after the war, were reached age 65, and both Alzheimer's disease and Parkinson's disease, for example, increase exponentially. The incidence increases exponentially after age 65. So there are a lot more people in the world with disorders associated with neurodegeneration, and so the need is high now, and it's only going to get higher. So what we want to do is have a drug which slows the process of nerve cell loss, and this has the potential to make a really big impact into people's lives and healthcare organizations around the world. So hopefully what this session will do is to throw some light into this area and try to understand where the next breakthroughs are going to come from. So what I want to do now is to go around the team that we've got set up for you, some leading figures in the field, First of all, I wanted to, and what I want to do is if they can introduce themselves and their companies briefly, and then we can go through a series of questions which I'll ask the team. So first of all, I'd like to introduce Richard Pither, who's the CEO of Cytox and is based in Oxford, in England.
Thanks, Alan.
Richard.
Yes, I'm Richard. I've been running Cytox for the last 8 years, and we're focused on the development of polygenic risk scores for identifying those at greatest risk of Alzheimer's disease and associated cognitive decline. And obviously, it's a very non-invasive test using a DNA sample, which can come from blood or very conveniently from a mouth swab. So you can actually send kits to people in their own home without having to bring them into a clinic to make that assessment. I've been working in Alzheimer's disease personally for over 20 years. Before taking the reins at Cytox, I was global head of R&D for GE Healthcare, and was my group was leading the development of the amyloid PET imaging agents in the form of flutemetamol, which is now commercialized as Vizamyl. And of course, that's got a very strong University of Pittsburgh link, because effectively it's the fluorinated version of PIB, which was developed by Bill Clunk and Chet Mathis in Pittsburgh. And our genetics actually very heavily relies upon collaboration with John Hardy at UCL, and also the Cardiff team of Valentina Escott-Price and Julie Williams.
Yes, I remember Bill Clunk telling me about this wild idea of using Congo Red to use in PET imaging studies.
Indeed.
We now move over to Switzerland and Dirk Beher, who's the CEO of Asceneuron. So over to you, Dirk.
Yeah, thank you very much, Alan, and welcome everybody. So I've spent 28 years in this field now, starting from my master thesis. So I've worked also in big pharma companies like Merck, Amgen, and Merck Serono before really founding this company like about 9 years ago. What Asceneuron is doing, we are really focused on the intracellular part of disease, so intracellular proteinopathies. So anything where you see intracellular protein aggregation. And what we're looking at really is the most common form of dementia with Alzheimer's. You have the tau protein in the neurons aggregating, and this really nicely correlates with the decline in the clinic, so much better than beta amyloid. Also, beta amyloid is clearly very important based on the genetics. The discoveries mentioned before by John Hardy really laid out the foundation there. The other one is also when you look at other diseases, we're looking at movement disorders. So in Parkinson's disease, you have alpha-synuclein aggregating as well. Lewy bodies, they're mainly made out of alpha-synuclein. And I think what we also like is that besides these really large diseases, which require long and heavily powered trials, there is often diseases as well. So essentially progressive supranuclear palsy is a disease where tau is aggregating. You have multiple system atrophy or Lewy body dementia where you have alpha-synuclein aggregating. So you can actually pick also smaller indications. Just on the company, what we really have, we have a clinical stage pipeline, and we have a mechanism we are targeting specifically, which slows down or kind of prevents the intracellular protein aggregation. We have highly brain penetrant molecules. They're inhibiting a specific enzyme called organocase. And what is interesting is that the field and we as well have really accumulated lots of preclinical data showing that inhibition of this enzyme can prevent the pathology with respect to tau from forming, so tau tangles. And what we are doing in-house now, we have our recent data also on alpha-synuclein, and we're expanding this as we speak here. So to really make the bridge that we have an approach that is more widely applicable to intracellular proteinopathies.
Thank you, Dirk. So we now move away from Switzerland to Denmark and Rita Balice-Gordon, who's the CSO of Neurotherapeutics.
Thank you, Alan, and it's a pleasure to be here today with this distinguished group. As Alan mentioned, I'm the CSO of Neurotherapeutics, which is a startup company focused on the endolysosome pathway and novel targets to elucidate protein trafficking and the handling of misfolded proteins, which, as Dirk just very elegantly described, are a major pathophysiologic feature of many neurodegenerative diseases. The second focus of MUNA is on microglial targets and on the interactions between microglia and neurons that both initiate and exacerbate neural dysfunction. Prior to joining MUNA in May, so it's a very new company, as Alan said, based in Denmark, I was the global head of rare and neurologic diseases at Sanofi, working on therapeutics for multiple sclerosis, Parkinson's disease, ALS, lysosomal storage disorders, and a number of other degenerative diseases. Before that, I was at Pfizer as the head of the psychiatry and pain portfolios in the neuroscience research unit, and a longtime academic with expertise and interest in cell signaling and neuroinflammation. I have an adjunct professorship that I still retain at the University of Pennsylvania School of Medicine here in Philadelphia.
Excellent. Thanks, Rita. So from Denmark, we're now going to move over to Luxembourg, and I'd like to introduce Jens Schwamborn from Organo Therapeutics. So over to you, Jens.
Hi, thanks for the introduction, and thanks for having me here. Yeah, my name is Jens Schwamborn. I co-founded Organo Therapeutics, and probably also directly apologies for the bad sound. I have some background here. The headphones are not working, so it's a bit messy. Anyhow, so what Organo Therapeutics is doing, we use in vitro models focusing on Parkinson's disease. And particularly, we start with patient-specific stem cells, so with iPS cells, and we differentiate those in three-dimensional structures that in many aspects resemble the human midbrain. So we call them midbrain organoids. The cool aspect is that these midbrain organoids really, in many aspects, recapitulate the complexity of the human midbrain, as well as many aspects of the pathology in Parkinson's disease. So we really see a loss of dopaminergic neurons. We see the accumulation of protein aggregates containing alpha-synuclein, all kinds of things that before have been pretty hard to achieve actually in vitro. And now we use that model to develop new small molecule compounds that are neuroprotective, so that pretty much stop degeneration of those dopaminergic neurons. And at the moment, with a focus on the molecular mechanism of autophagy. I'm looking forward to the discussion here. Thanks.
Lovely. Thanks very much for that, Jens. So what I'd like to do now is move into question time. We've got a panel which has got diverse backgrounds and companies with different perspectives on the same problem of trying to identify and develop new medicines for neurodegenerative disorders. So I was wondering from the panel if they could sort of give us an answer to the question of what do you think the major advances have been in this area over the last 5 or so years?
Well, Alan, if you'd like.
Maybe I could kick it.
Yeah, go ahead, Rita. Ladies first.
Okay, so Alan, I thought you were going to throw the questions to us. Otherwise, we're going to be stepping all over each other. So apologies. But I thought I might kick us off just by saying that I think one thing that has revolutionized the biology of neurodegenerative diseases and has opened the door to a number of interesting, potentially impactful therapeutic approaches is the impact of human genetics, not only on Mendelian genes that cause disease. You mentioned APP and inherited Alzheimer's disease, but risk factors and SNPs in genes that confer either risk or developing disease or protection that have highlighted some common pathways of neurodegeneration that link together diseases as previously thought to be so disparate as ALS, for example, and FTD on a spectrum, Parkinson's and Alzheimer's disease sharing many common mechanisms, and have allowed us to focus on core cellular processes that could be corrected, not only in the context of a disease, but potentially then applied impactfully to other diseases as well. And this is kind of core principle is what led me to join Muna and what led Muna to focus on endolysosome pathway as the major cellular system for handling misfolded proteins and shuttling them to different areas of the cells to be handled. So I think to kind of wrap up my answer, the advent of human genetics that has allowed us to link previously very disparate processes to diseases that share common pathophysiologic etiologies has really been a major advance to the field.
Yes, thank you. I think one of the big challenges we have with brain disorders is their complexity and the fact that our understanding is far from complete. So yeah, that's a very good answer. Anybody else want to come in on that question? Richard?
Sure. Yeah. I mean, I would like to pick up on Rita's point about genetics, of course, because but from a slightly different angle, in the sense that this polygenic component that underlies a lot of neurodegenerative disease, I'm focusing on Alzheimer's disease, obviously, allows the earliest possible description of those at risk. and that means, whether you're recruiting for a clinical trial in pre-symptomatic people, or whether you want to confirm at the symptomatic stage whether you're dealing truly with Alzheimer's disease or not, then I think that they have tremendous value. And I think that really is something that has only emerged at a sort of consensus level over the last 5-6 years, really. That Alzheimer's disease, that late onset Alzheimer's disease, actually does have a major genetic component. I mean, you could go to conferences on Alzheimer's 7 or 8 years ago, and the genetic session was the last thing on Saturday morning, and most of the people had gone home by then. Whereas now it's up front and center because of this real change in understanding. So I think that's one thing I would highlight. The other, though, I think is the early detection from a diagnostic standpoint is really useful for the individual. I'm thinking of from a patient perspective now, if there's something you can offer them. And I think what's, again, changed incredibly in our field over the last few years is the fact that healthy lifestyle, risk avoidance and management can actually have a major impact on the severity and the time at which symptoms might develop. So I think Mia Kivibelta's team in Finland, the so-called FINGER study, has really pointed the way to showing that individuals will benefit from understanding where they are on the risk spectrum, even if they turn out to be on the high side, because then they can go in and manage proactively their own health outcomes. And I think that's a tremendous positive benefit to the field while we still await meaningful disease modifying therapies. So I think I would pick genetics and health and lifestyle as two major advances over the last few years.
Yeah, there's not much we can do about the genes we have, but there are other risk factors which we can modify.
Precisely so.
The other thing is that the work from Brack and Brack in Germany have shown, look where they looked at hundreds of post-mortem brains. From their data, it looks as if the disease, Alzheimer's disease, starts as many as 42 years before symptoms appear. So having some risk assessment of some assessment of the risk of somebody developing Alzheimer's disease, and knowing you can modify it, is definitely a good way to go.
Precisely so, and I think genetics can identify those at risk, and then the more biodynamic markers can pick out those where that risk has become manifest at the level of an ongoing process, which would allow more appropriate intervention. So yeah, I think those things are absolutely linked.
Yeah. Any more?
Yeah, maybe I add one thing. It has to be, I think, Richard, is probably your in your shop anyway. In the past, it's the imaging agents, so that we can actually now identify the right patients for the right drug. We can stratify, at least in Alzheimer's. We have agents for beta amyloid. We can say, okay, at what stage the patient is presenting. Do we want the people when they have amyloid, or do we rather want people in the trial they have hardly any? And I think the tau imaging agents are evolving as well now. And I think this will hopefully spill over into the rare diseases as well. As you know, these tau imaging agents at the moment are not suitable for repeat tauopathies, where we have the rare diseases, most of them located, so that we can ideally at some point, say, okay, we're running a trial here. We want people with 4R, 3R or mixed pathology, or we want synuclein, or we want TDP-43 or whatever, depending on the drug. And I think that's something, we couldn't do 20 years ago. I think these Alzheimer's trials, they're mainly based on cognitive, MSSE or MMSE or whatever classification. And there was no idea, what kind of pathology was already there when the trial started. And that probably has led to many failed trials, I have to say, is my view, because we didn't have those tools, and we also didn't have the right molecules to test in the clinic either.
Yeah. Well, the discovery that's got me excited is the interference RNA in Huntington's disease, because that's got the potential of completely changing our field if they get a positive result there, which would be good, because I think it'll bring a lot more interest and investment into the neuroscience arena. That's good. Okay.
Raise my hand for the cell biology in that context. So I think that these technologies that Yamanaka developed, so to reprogram a somatic cell into an induced pluripotent stem cell, which really allows us now to do patient-specific personalized approaches, was a game changer. I think in the academic as well as in the biotech world, there's so many approaches now based on iPS cells that allow us on the one hand to access the genetics, but also understand probably things beyond genetic, because we can look at phenotypes derived from those iPS cells. So, and this is also obviously the basis for our work at OrganoTherapeutics. So I think that was a game changer.
Yeah, excellent. Okay, good. That's good. The next thing then is animal model. Let's follow the animal model side of things. So what do you think is the gold standard in terms of animal models, or do we have a gold standard? And I think when it comes to animal models or cellular models of disease, the key measure for me in terms of discovering new medicines is how predictive those models are. What's their predictability? And the whole field doesn't have a good record in that regard. I think one of the few examples is in the head injury arena, because when I worked in the Head Injury Research Center in Pittsburgh, we showed that if we cooled experimental animals down to the core body temperature of 32 degrees for 4 hours, the amount of damage after traumatic brain injury was markedly reduced. And then we, with clinical colleagues, we translated that into patients with severe head injury, cooled them down to core body temperature 32 for 24 hours. The amount of their recovery was much improved, and that's now become a standard measure of care. So that model predicted efficacy in the clinic, but there are not many good examples of that happening, and which is a problem, because without having compounds which show efficacy in the clinic, it's very hard to validate a preclinical model. So we've got a catch-22 there. Does anybody want to pick up on that?
Yeah, maybe I start, because we do still a lot of this work and we are banging our heads here. You know, it's a very good question. So I think my view is, to be fair, we don't have a gold standard model. I think what we have is models of pathology. You know, we have a mouse that overexpresses mutant tau, then you get tangles, and then you get some deficits, but it's not even clear whether deficits are really due to the tangles, because you know, there's this very famous TG4510 mouse line. There was a recent paper that some of these deficits are actually integration of the transgene into other genes. You know, it's not even the pathology itself, but it matches up. So I would say what we have is models where we can measure that the drug changes the progression of the pathology very well. I think the gap to efficacy and behavioral benefits, that's still there. I think there's some models where we see some connection, and those are the ones we're using, where we actually have a better confidence that the pathology is at the right place and the deficit is the right place. You know, in Parkinson's, we have some synuclein overexpressing mouse models. They give you motor deficit, and I think you have a link that it's really caused by the overexpression of alpha-synuclein. So that's one example, for example, we are using as well. So I would say that's the thing where we know we can translate into the clinic. And then what we will have to do is we really have to think hard, you know, what are we going to do in phase 1? What are we measuring, ideally already in patients? What is predictive then of the larger scale treatment? You know, and I think this still has to be done in humans, because to be fair, you know, I think in neurodegeneration, most of the models are pharmacodynamic, you know, pathology driven. It's my kind of view here.
Sure.
I could jump in here and say I completely agree with Dirk. I think the models are most useful to prove biologic mechanism and to ask questions about mechanism. But we are, I think, as a field, guilty of using models to ask questions that they're not well suited for with perspective of recapitulating all of the features of a disease. And I think once you've used a model to establish the biological mechanism, then the second thing that models are good for is to establish target engagement. And that doesn't really have to be a genetic model. That could be a wild type animal. And then I agree with Dirk completely. The faster one can establish safety and get to phase 1 and establish safety in humans, you can then explore early signs of efficacy in phase 1b or phase 2a trials that allow you to really assess the mechanism in the context of the patient and their particular disease setting. I think the faster we do that and the more we do that, the better off we're going to be. I happen to think we tend to linger too long in discovery and preclinical phases, and we do too little in phase 1b and phase 2a, and this has hampered the field and will continue to hamper the development of impactful therapeutics until we figure out a way to test and retest in phase 2 to really identify therapeutics that are going to have a real world impact in patients.
Yeah. Jens, do you want to come in there?
Yeah, I think my view on the animal model is a bit more radical. I think we should put them as late as possible in the preclinical pipeline. Probably if we talk about the rodent models, really more like for side effects and tox, get as much data as possible with relevant human models, and then probably do a final validation in the primate model. But I think we have a huge problem when it comes to neurodegeneration with rodent models. So I would advocate for replace them as much as possible.
Yeah, I recall the 1980s where I did a lot of work in the stroke arena. Then the predominant hypothesis at the time was the excited toxin hypothesis, that excessive glutamate was causing neurodegeneration. There were a number of pharmaceutical companies that had antagonists, particularly NMDA receptor antagonists, and they went into a number of clinical trials and they failed. And people thought it was because of the disease models weren't any good. So I took 17 or 18 of those compounds and did a simple study. I found out the minimum effective dose to cause efficacy in a stroke model and the minimum dose to cause side effects, looking at the motor coordination, the road test, and discovered that nearly all of the compounds had a therapeutic ratio of 1. Some of them had a therapeutic ratio of less than 1, which meant that you got side effects before you saw any efficacy. So I think one of the big problems was that particularly in the US, where they were in fear of litigation, they kept the dose down below the, to avoid side effects, kept the dose down. So it was oftentimes a sub-therapeutic dose that was given. So, the safety issues that you mentioned are critical as well, and that certainly needs to be taken into account. But what do you think in terms of looking at PK/PD relationships as a surrogate of as a measure of or a replacement, or using instead of an animal model?
Yeah, I think you establish your PK/PD on the target and the receptor occupancy or enzyme occupancy Rita was referring to. That's very clear. But you still like to see that you see something on the intended biology. So I would say you still want to show, example, you have a synuclein modifying agent on an enzyme. You want to occupy the enzyme. Your dose and drug has to get into the brain. You need to have 80 or 90% coverage. And then you want to see something on the pathology. I think there's still, you need to bridge, so to speak, to say, okay, here's our target, here's our mechanism. But we need to really see something happening as well on the intended pathology. And then I would say you have to define a really smart phase 1b, where you can show something in patients, either in CSF or plasma, because then I think that's one of the things that have changed now as well, that we're much earlier thinking about, how can we translate that biology as early as possible in the clinic? In the old days, it was like, okay, if you do your phase 1, phase 2 is just a dose range finding, and phase 3 is really where you probe, and then you have spent a lot of money already. And I think this is what most companies are now walking back, saying, no, we put the hurdles up early to really show in phase 1b what you can show before you make all this investment and commitment to go into these larger trials.
Yeah,
I think that's a fair point, Dirk, but I also think there's a danger in being driven by pathology-led hypotheses. That's been the problem for the last 20 years in Alzheimer's disease, where the focus has been primarily on amyloid and then more recently on tau. What we know is that these things form and aggregate in the brain. What we don't know is whether they're sort of cause or effect. And the fact that we've had 20 years of failure in drug trials seems to suggest that these models are sort of self-fulfilling prophecies, in the sense that, yes, they show you can remove the target protein, but they don't show you can do anything about the underlying cause of the symptoms and the disease. So I think there are two lessons for this. I think, first of all, we have to continue to invest in very basic research, mechanistic research for all these complex degenerative diseases, because I don't think we really understand as much as we would like to believe. And just because you overexpress a target protein in a model and then you can remove it again, it doesn't really tell you anything about the effectiveness in the clinic. And I think in Alzheimer's specifically, one ray of hope, I think, is the fact that the relative emphasis on amyloid is moving. It's reducing. There's, a few years ago, it was like 60 or 70% amyloid targeted therapies. Today, it's about 35% of the drugs that are in clinical stage development at the moment, and a lot of other mechanisms around neuroinflammation, synaptic biology, vesicular transport, neurogenesis. These are coming in now to Alzheimer's, relatively recently, and I think that's a good thing. But I think Alan made the point at the outset that until you understand, you know, you see, until you've got some drugs that show a little bit of efficacy, it's quite hard to know what an appropriate model is really for testing these drugs beyond sort of the toxin and safety elements.
Yeah, that's right. There's definitely an element of confirmation bias in the approach. I'm reminded of a study that we did when I worked with David Bowen. We collaborated with a group in Manchester, looked at Alzheimer patients that tissue removed a biopsy, and that allowed us to relate all of the patients were assessed psychologically, psychometrically 2 weeks before the surgery. And then we looked at the correlations between number of plaques and cognitive impairment, a number of measures of cognitive impairment, number of plaques, number of tangles, and also a number of pyramidal cells in layers 3 and 5. The plaques didn't correlate at all. The tangles showed a weak correlation, but the best correlation of all was the correlation with the number of pyramidal cells lost in the temporal cortex. And so, you know, that from that data, you know, it was suggested that the key determinant is loss of these pyramidal cells, and pyramidal cells are the most abundant neuron in the cerebral cortex, and they're the cells responsible for communication from one area to another. And so, you know, and that may be, you know, maybe because of the formation of tangles, or it may be some other mechanism we don't know. But to me, certainly,
I think the other point is, Alan, that we kind of talk blithely about Alzheimer's disease as a single disease, and yet, you know, there are clearly different manifestations of it. You know, and if you look at the symptomatic level, you see differences there, but you certainly, the underlying biology seems to be different. And there are publications now suggesting there are at least 3 different subsets, and therefore maybe expecting one drug to work equally well across those subsets may be a bit hopeful. And I think, again, you touched on this earlier when you mentioned the acetylcholinesterase inhibitors and the symptomatic treatments. I mean, there's a huge range of responses to those. You know, these are generics, they're cheapest chips, and yet for some people, they have a huge benefit. I mean, I can speak from personal experience. With my mother was diagnosed earlier this year with Alzheimer's disease, and given memantine, and given what I knew about memantine, I was very, you know, unexcited about that. And yet it's transformed her life, you know, and that's almost 12 months into the, you know, the start of that treatment. So I think we should, you know, take the chance really to reflect on, you know, this kind of pleiotropic nature of all the diseases that we're working on, and then ask really whether, you know, or one shot type mechanism, anti-amyloid, anti-tau, whatever it might be, is really the most appropriate.
So I think Richard makes a very good point. I want to amplify 2 aspects of it. The first one, just as a scientist and an academic, I think it's going to be interesting when the history of Alzheimer's disease is written to reflect on how the single-minded focus on the amyloid hypothesis really slowed down, I think, an appreciation of exactly what Richard said, that Alzheimer's is a spectrum disorder with many drivers, likely many drivers, and that, you know, focusing on a pathology that may be the effect rather than the cause has really slowed us down. So that's the first thing. And then the second point I'd like to make, also amplifying what Richard has just said, is that we've all been focused on disease-modifying therapies, right? Slowing or preventing neurodegeneration, really changing the course of disease. And that's a very noble goal. But symptomatics may offer some low-hanging fruit for impact to patients. So Richard mentioned memantine. There are drugs in clinical development that address sundowning or aggression, anxiety, depression. These are symptoms that may not excite us as cell biologists interested in disease-modifying therapies, but are incredibly important for patients and caregivers. And I'm very pleased to see really across the globe a renewed interest in applying our efforts to developing impactful symptomatics that may prevent entry into managed care by as little as 3 months, but would still be tremendously impactful, not only to patients and caregivers, but to healthcare and economies. So I think this is another area that deserves really careful attention.
Yeah, and I completely agree. And one of the symptoms that leads to the institutionalization of patients is the psychiatric symptoms. So that's a massive unmet need there. Okay, lovely. Any more there for anybody? So moving on to the Alzheimer clinical trials, the history is that they failed to show efficacy, and then what happened is that the pharmaceutical companies started looking at earlier stages of the disease. So we went from severe to moderate severe to moderate mild, and then just mild, and even mild cognitive impairment. And this is where biomarkers come to the fore, particularly sort of Richard's approach. But do you want to comment on segmenting and patient populations in that, and how we can sort of enhance the clinical trials by having a more homogeneous population by use of biomarkers in this area?
Sure, yeah, I'll start, and maybe I think Dirk's probably got a perspective on this as well from some of his earlier comments. But yeah, I mean, I think this is one of the big leaps forward. I mean, we learned, well, over 10 years ago now, where Janssen and Eli Lilly were basically testing their bapineuzumab, solanezumab anti-amyloid therapies in subjects who turned out really not to have the target protein in many cases. So that kind of spawned the requirement to look more closely, using both a combination of PET and then CSF, and today even plasma measures of target presence, amyloid and tau fragments in particular, but also NFL, neurofilament light, as well as a measure of neurodegeneration. So I think these things really are a huge step forward for the field. The problem is accessibility and robustness, I think, of those tests. So, PET, certainly the earlier you go in the disease pathogenesis, the lower your hit rate will be to find a PET amyloid or a tau positive individual. And if that test is going to cost you $5,000 to $10,000 each time, that's an awful lot of money wasted on people that won't have the target. So what options do you have? Well, you could do a CSF test, but that's very invasive. A lot of certainly pre-symptomatic individuals don't like lumbar puncture. A lot of physicians don't like giving lumbar puncture because of the associated risk. So so that's moved the field towards looking at serum biomarkers. The problem here, of course, is one of very low levels of analyte and not very robust and reproducible measures, because a lot of the fragments, particularly amyloid fragments, are very hydrophobic. So the way you take and handle the sample, whether you leave it on the bench for 10 minutes too long, or even diurnal fluctuation, I mean, all these things can make a huge difference to the actual quantitation of these analytes. So it's not easy. It's easy to say, but far less easy to do in practice. And there still today are only probably a couple of labs in the world that seem to be able to really reproducibly carry out these tests. So that's where, I'm just to ring our own bell for a moment. I think that's where genetics does come in, because by definition, DNA is a very stable analyte. You can dig up a woolly mammoth that's been in the ground for 4,000 years and sequence its DNA still. And it means you can send a kit out to someone's home and they can abuse it all they like, but when you come back, you've still got something you can measure reproducibly. So it gives you a way to then stratify people into more invasive techniques that are more expensive and potentially more hazardous. So I think, yeah, for sure, the biomarker field will make a huge difference, not just in Alzheimer's, but across neurodegeneration, in making sure we're getting the right patients into studies. And then beyond that, to look at response and non-response and try and work out multi-component definitions that would allow you to then triage and stratify more appropriately into subsets for additional phase 2 and phase 3 studies. So transformational potential, I would say.
Excellent. Dirk?
Yeah, I think I don't have too much to add. So Richard nicely summarized it. I think what I liked a lot is that these efforts of Diane and Adney helped us to understand about the temporal relationship of these markers. When do they come, which one is first, tau, amyloid, etc., neurodegeneration, when do these things happen in the time course? And I think I personally like the plasma biomarkers now with tau 181 on these kind of things, because they have shown a really nice correlation, and they should be technically more easy to handle. I fully agree. CSF A beta is really tricky. Even the number of samplings you do in a patient will determine kind of your signal. So that's for the experts. But I think overall, I think this is really the way forward. And we shouldn't forget, in Alzheimer's, people have worked this out to a certain extent. But when you go away to more rare diseases, there's nothing known. So when we look at progressive supranuclear palsy, we are very interested that there's hardly anything known, because there's not enough patients, for example. So I think there's still a bit of work to be done going forward.
Yeah, Jens, do you want to say something about the Parkinson's side of things?
Yeah, I pretty much agree to what has been said. Genetics certainly is going to be the key. For Parkinson, we have probably the luxury that there's at least a certain subpopulation of patients that have a clear monogenetic cause. This could be the entry points for something. And then further on, it's going to be very interesting to look at polygenetic risk scores. So if we get really whole genome sequencing of individuals, we're probably going to be able to really substratify into groups that are maybe more autolysosomal, more mitochondrial, more protein aggregation type. And with this genetic information, we could probably indeed define better cohorts for clinical trials. And probably even some of the things that have been failed in the past would work if they would have looked at in the right substratified cohort.
Sure.
Maybe I can just amplify what's been said already by adding that there's so much work on biomarkers across the field in many diseases, not just CSF and serum, but saliva, tears, urine. It's very difficult to think of a program that actually gets to the clinic that doesn't have a biomarker strategy. So there's a ton of data, not just for Alzheimer's and Parkinson's, but being collected from humans, and the ability to use machine learning and sort of multi-omics analytics to assess biomarkers, not only of disease progression, but of therapeutic efficacy. This is going to be a very rich source of all-in-human data for the next, well, for the foreseeable future. It certainly has been already, but I think it's going to be key to translating what we do in the lab to the clinic for some time to come.
Excellent. Okay, good. Well, and then moving on to another question then is in terms of where the field is, because we're in the midst of a COVID world now, and infectious diseases have come to the fore. Cancer has dominated oncology has dominated the focus of many pharmaceutical companies. So where do you see sort of neurodegenerative disorders within the remit of biotech and pharmaceutical research in the years ahead?
Well, maybe I could kick us off here, having worked both in biotech and big pharma. I think there is a tremendous amount of innovation and creativity and frankly risk-taking in biotech arena and neurodegenerative diseases, and I think some of the best science is being done in biotechs and being translated from academic labs to biotechs, where I think big pharma companies are much better at the execution phase when it gets to the clinic, in particular phase 2, phase 3. But my best guess is the first success that is kind of a home run success in neurodegeneration, whether that's AD or PD or ALS. I think big pharma companies will be back in, and that renewed investment and interest will help fuel sort of the next cycle of innovation. I think there is a misperception that there are other therapeutic areas beyond neurology, like oncology and immunology, that are low-hanging fruit for drug development. I think that is mistaken. And I think people conflate broader neurology with Alzheimer's, where, as you said, the history of clinical failures has really left a kind of a pall on the field with respect to pickup by large companies. But I am very encouraged by the creativity, the dynamics in the biotech arena in this field, and I think it's going to carry the day.
Sure. Any more? Dirk?
Yeah, I think it's fair to say that in Alzheimer's, we have more experienced headwinds than tailwinds. You know, that's very clear with the failures. And I think I can only second what Rita was saying. You need one success, one big success, and every company will walk into this. I think I'm not an oncologist by training, but I've seen immuno-oncology being talked about 30 years ago, and nothing worked. And then suddenly there's one hit, and every company needs to have a PD-1 antibody or a PD-1 ligand antibody, etc. So I think for us, I think if I look into the near-term future, I think the approval or whatever happens to this aducanumab will be quite important, because I think you can debate, you know, whether it's efficacious enough or not, but it will influence the field, you know, because if there is an approval, there will be some positive things, and if not, it will be negative. And I think I like one of the comments I've seen one of these public forums, this in other diseases, that's exactly how it worked. You come with a drug that isn't terribly efficacious, but there's something happening, and then you add more and more. I'm also a believer that we, at the end of the day, need a combination therapy, to have multiple things addressed, and it could be symptomatic together with the disease modifier or two different disease modifiers, etc. So I think what I'm certainly looking forward to, and I think is something that gives us really a tailwind in the near-term future. And I fully agree also that there's a lot done in biotech. We are close to academics, we are close to more risky things, and that all ultimately leads to innovation. I would say that's the mechanism there.
Sure. Thank you, Dirk. Richard?
I would agree with much of that. That I think aducanumab, getting some kind of limited approval would be a great fillip for the field, and because we're all driven, whether we're in big companies or small, by investment availability. And today, if I'm looking at, as an investor, looking at oncology versus neuroscience, I'm looking at roughly a 20% success rate in oncology trials versus 1% in neurology, neurodegeneration. So where am I going to put my money? It's an obvious answer. That's why I think there is a real role for government funding here, and organisations like the Alzheimer's Association and the European equivalent do a great job, but they need to do better, because there's still too much of a lopsided investment in oncology versus the trickier sort of neurology side of the equation. But I think if we've learned anything from COVID, we've learned that where there's a huge health and economic threat, that people can actually work incredibly innovative ways to solve these sorts of problems. And so I think it's beholden on us to sort of continue to explain the fact that Alzheimer's neurodegeneration generally is a disease of older, aging demographics, and that's where we all live in populations that are living longer and getting older. And the economic hit is huge. I mean, we're measuring this in trillions of dollars. So I think there is a sort of an urgency, a kind of burning platform there that we need to keep explaining to those who can pump prime, the funds that will get these things moving, in a more sort of successful way in the future.
Thank you. And for clarity, aducanumab is the monoclonal antibody from Biogen, which is under review by the FDA at the moment, and we expect to get a readout first quarter of next year, I think. So, yeah. Jens, anything from you?
Yeah, I think there could be some support coming actually from an unexpected direction. So living now in this COVID-19 world, there's more and more data accumulating that this virus probably even has an impact on the brain. So I think it shifts also the attention to something else. There's data showing that, for example, in the COVID model systems, tau is accumulating. We have these data showing that amounts of Parkinson cases after the Spanish flu increased significantly. So I think that could be a new trigger for investment going into that direction.
No, I think you a very good point, Jens. Very good point indeed. That the long COVID has many neurological psychiatric consequences, and they're only just becoming clear now, and it's going to require a huge amount of research and effort to tackle this issue. It's essentially a new disease that's being created, isn't it? Which hasn't really been defined properly yet. Yeah. Any final comments for everybody before we wrap up?
I'd just say one thing, Alan. I think that to work in this field, you have to be an optimist by nature. Otherwise, you would get fed up with banging your head against a brick wall. So I think the reason to be optimistic is very much, as we said at the outset, the fact that people, individuals, are desperate to know about risk and how to manage it. There's a real demand there, and I think that will drive action on the part of drug companies on the one hand, charities and government organizations on the other. So I think that work that I highlighted at the beginning from Mia and the finger study is for me one of the most important drivers that we've seen in the last 10 years.
Excellent. Any more?
I'd like to just add that I'm encouraged by how many really smart and committed younger scientists want to get into neurodegenerative research, whether it's an academic setting, biotech, pharma, there's a great pool of talent out there. And one of the other side effects of COVID that we were laughing about as we started this call is that COVID has really highlighted the impact of science and scientists. And I think this impact will carry forward to other areas of science as well. But the influx of talent and the committed nature of that talent is really a tremendous resource we'll be tapping for the
I think you're
years
right.
to come.
What COVID has done is highlight the relationship between science and society. I think it behooves all of us scientists to make more of an effort to communicate our science so that the lay audience can understand it much more easily than they do at the moment. So we have a better connection and increased confidence in science and neuroscience in particular.
Just to add, I think this was mentioned before. I think I'm encouraged by the fact that we're not just following one hypothesis anymore.
No.
We're doing many, many different targets. And I have to also say the way we're doing it has improved, We're not moving drugs into the clinic that don't get into the brain and not these kind of things, I think we are more selective. And I think that helps at the end of the day, test more higher quality molecules, and then hopefully we get also better readouts in the long term. So I'm encouraged by that as well.
Trying to develop compounds with a therapeutic ratio of 1 or less, exactly, yeah.
Yeah.
We've learned lots of lessons along the way. Jens?
Yeah, I said before, I think you have to be an optimist in this field, and I'm pretty optimistic. I'm particularly looking forward in moving into this area of personalized medicine. I think that will change a lot of things, how we see diseases, but particularly how we treat them.
Exactly. Well, it's good. Well, I think that brings the session to a close. I think hopefully that's given you all a good overview and sense of the field of discovering new drugs to slow the progression of nerve cell loss in the brain through chronic disease. So I'd like to thank you all for your attention and all of the contributors for their excellent presentations and interactions. Thank you very much. Yeah. Tony, over to you.
Thanks,
thanks, Alan. It's always nice when I listen to such a fascinating conversation and I get to do the easy bit of popping back up at the end to say thank you, and nothing more is expected of me in terms of insight. But I can just thank you on behalf of One Nucleus for contributing to the content for Genesis 2020. I think some of the points you raise give us grounds for optimism, and I think that's all we can really hope is to enthuse people to say keep believing in science and keep enabling you guys to do what you do best, and that's come up with better patient outcomes. So thank you very much. Thanks for joining us from various parts of the globe, and I hope to see many of you soon. Thank you.
Okay,
thank you.
Thank you.
Thank you very much. Thanks, Alan.
Bye-bye.